I'm currently working on the development of technologies for the fast identification of clinically relevant T-cell receptors. So what we are doing, on the one hand, we are developing functional screening that are based on TCR avidity, which in other words is the binding strength of a TCR towards the cognate image seen. And what we did was to implement this functional screening into the protocol and the pipeline for isolating T cells that express T cell receptors specific for a certain antigen. So at the end of the day, what we can achieve simultaneously is, on the one hand, to get the sequence of the T cell receptor that can recognize the antigen of interest, and on the other hand, we can also predict the functionality of this T cell receptor. And this is very important because we can have an educated decision on which T cell receptor to re-express in later stages for functional screening. This is crucial because this, functional characterization, because this functional characterization is really time and cost consuming. So in parallel to the establishment of this functional screening, we are also setting up approaches for increasingly faster screening of T cell receptors in order to narrow down the list of candidates to only few lead candidates for clinical application. And then last but not least, we are also optimizing protocols for T-cell engineering, and in particular, we are focusing on non-viral CRISPR-Cas9 mediated T-cell engineering. We developed a protocol for a complete replacement of the endogenous T-cell receptor with the transgenic TCRs, and we could show that in these regards we can obtain T-cell products that are near to physiological T-cells, and they should be safe, with a safer profile and also more reliable in terms of functionality.